Hello scholars, today in this video we are going to look at two tests that we perform in the lab, the nose and the throat cultures using the mannitol salt agar and the blood agar respectively. Let's start with the nose cultures. The nose culture is done on mannitol salt agar. This particular medium is selective and differential for Staphylococcus aureus. It is selective because in the ingredients it contains salt at a concentration of about 7.5%. This salt makes the medium selective because its concentration is high enough to dehydrate and kill most bacteria. Staphylococci, though, they thrive on this medium, largely because of their adaptation to salty habitats like the human skin. We will be swabbing the nose to get our culture. Now, the medium is differential because it helps differentiate between the different species of the genus Staphylococcus. It contains mannitol, which is a Staphylococcus aureus favorite. And if it ferments the sugar, then the phenol red that is within the medium would turn yellow because the pH will drop to acidic below 6.8. It will remain red around 7.4 to 8.4, and it turns pink at 8.4 and above. The mannitol salt agar is used for isolation and differentiation of Staphylococcus aureus from other Staphylococcus species. It is not used to determine the ability of an organism to ferment the mannitol. As far as the procedure goes, you will take a swab from the nasal cavity. The mannitol salt agar would be inoculated using the quadrant streak. The very first quadrant, quadrant 1, the cotton tip swab is used, and it is applied using that back and forth for the first quadrant. Then for the second, third, and fourth quadrant, we switch to the loop, and that's where we will continue with the quadrant streak. Then the plate is incubated at 37 degrees overnight. This figure is showing the mannitol salt agar in its uninoculated state. And then Staphylococcus epidermis is added. It can grow because it is well adapted to growing at high sodium chloride concentrations. But this medium, like I mentioned before, will be able to differentiate between the different species of Staphylococcus because if it is Staphylococcus aureus, it will use the mannitol and turn it yellow, as you see in this portion of the plate. When this is done in class, about 20% of the students will carry the S. aureus. So if we do this and we look at the plates in class, out of the 25 students that we have, about 4 to 5 of the students would get a positive result. So let's look at results that are more likely in the classroom. Now, this is an artificial setup of the plate, but let's look at results that are more likely in the classroom. When we look at our nose culture results on the mannitol salt agar, most of the plates would look like the one that's before you right now. This is considered negative. In this plate, the organisms that grew were not able to ferment the mannitol into acid end product. But in about 20% of the students, we will see a plate that looks like this one. This would be considered positive, or let's call it a suspect positive. If someone gets a result that looks like this, there is a second step that will need to be carried forth, because even though it is turned yellow, it does not necessarily mean that for sure it is Staphylococcus aureus. So we want to confirm that. In step 2, we will take one of the yellow colonies from the suspect plate and we will inoculate rabbit plasma and incubate it at 37 degrees Celsius. If the rabbit plasma clots, as in the top test tube, then we are certain that this is Staphylococcus aureus. If it does not clot, as we see here, this is still liquefied, then no further testing is needed. But if it clots, we will require further testing. We'll have to move this to step 3. Now, the medical significance of testing for the Staphylococcus aureus would be determining if someone is infected with MRSA, for example, methicillin-resistant Staph aureus, or maybe VRSA, vancomycin-resistant Staph aureus. That's what the significance is here. So now we will take it to the step 3 just to check. Let's make sure that this Staphylococcus aureus is not antibiotic resistant. So we're going to do the antibiotic sensitivity test, which we have already covered in class, where we use those plates called the Mueller-Hinton plates, and we dispensed discs that contain different antibiotics to see if it was either susceptible or resistant to that particular antibiotic. You will only move these tests forward if in your first round of testing, you got yellow colonies and it turned the medium yellow. If it did not turn the medium yellow, if it was negative, then there is no need for further testing. Our next test on the agenda today is the throat culture, where we would be using the blood agar. Several species of gram-positive cocci can produce exotoxins that we call hemolysins, which are able to destroy red blood cells and hemoglobin. The blood agar has about 5% sheep's blood within a tryptic soy agar base. So this particular medium would allow for differentiation of bacteria based on their ability to hemolyze or break down the red blood cells. In order to perform this test, students will pair up and each would take a sample of the other partner. You want to avoid touching the sides of the mouth and the tongue. The best technique is just to take the swab, depress the tongue, and go straight back towards the back of the throat. The application here is that blood agar is used for isolation and cultivation of many types of bacteria, and it differentiates them based on their hemolytic characteristics, especially within the genre Streptococcus, Enterococcus, and Aerococcus. The thing is, with the Streptococcus, this is a test that we would use to check for S. pyogenes, which is the causative agent of strep throat. The three major types of hemolysis are alpha hemolysis, and in alpha hemolysis, there is a greenish zone that occurs around the colonies, and this is because of the incomplete lysis of red blood cells. Next, we have gamma hemolysis. This means no hemolysis. I like to think of the G in gamma as good. This is what we want to see when we take a sample from the patient and inoculate the blood agar. And then there is beta hemolysis. And in beta hemolysis, we will see a complete lysis, a complete breakdown of red blood cells. And I like to think of B in the beta as bad. We do not want to see this. And this usually indicates strep throat from S. pyogenes. In a class of our size of about 25 students, we will get 0 to about 1 of the beta hemolysis. And typically this student is already complaining of a sore throat or that particular symptom comes on within the day. For the alpha, we probably would get about 2 to 3 students that will exhibit the alpha hemolysis. And in the gamma, that would be the majority of the class, so almost everyone else. Now this wraps up the 2 labs that I wanted to cover in this video today. Of course, if you have any questions, you can feel free to contact me. And in the meantime, happy studying.